So in the last lesson, we looked at the cell and the organelles, or the organs of the cell that are found within. But what we're going to focus on now is what surrounds the cell and keeps it in its particular shape and helps it allow things in and out. So at the top, I've got quite a complicated image of the cell membrane. And what we're going to do in this lesson is just break down what each of these components are. So before we can talk about the structure, we need to talk very loosely about the function of the cell membrane. There are four main ones, and they're definitely worth knowing. So our first major function is that the cell membrane, similar to your skin in a way, is a physical barrier. Okay, it's what separates the inside of the cell from the outside of the cell, and it's what allows the cell's internals to be different from its surrounding environment. Secondly, it's going to control what materials can come in and come out. Think of it like a guard at the door. It's effectively going to allow certain things in, and it's also going to push certain things out. So things like waste products get out versus really useful things like maybe oxygen or different ions like sodium in. In terms of sensitivity, what I mean by this is a really important word, and it should be a word if you're ever asked to describe the function of the cell membrane that you use. This is effectively the cell membrane's job in detecting any changes that happen in the external environment, be it a change in particular ions concentration, or perhaps even detecting a foreign cell which could be pathogenic in nature. Finally, the cell membrane itself is attached to the cytoskeleton. Now, we briefly mentioned the cytoskeleton in our lesson on cell organelles. Again, it's just the skeleton of the cell. It's what helps to support and provide some structure to the cell itself. So now that we've had a go at describing some of the functions, let's talk about the cell membrane itself and what that would actually look like on a day-to-day basis. Now, something that's commonly used to describe the cell membrane is this fluid mosaic model. Now, all this means is fluid, I want you to think of something that is constantly moving, okay? It's changing position, it's on the move, it's never staying perfectly still, okay? And when we're talking about mosaic, all that we're saying, it is made up of many different molecules. And these are the things that we're going to talk about in this lesson. So phospholipids, proteins, carbohydrates, and cholesterol. And we can see that in this image on the right. We can see that the cell membrane is not just made up of one thing, it's got a lot of smaller components within it. But the thing that we also need to talk about is not just that mosaic, we also want to talk that all of these things are constantly moving and flowing. So what we have is a diagram, and in this diagram what we can see are all of these different components. We're going to use this a little bit later on to help us label this blank one here, but before we do, let's learn a little bit about these individual components. So, phospholipids. The key part of phospholipid is this lipid component here. Lipid is a form of, oh, that pen's not very good in this situation. Let's go to green here. This lipid is effectively a form of fat. Now, these fatty molecules don't really mix that nicely with water, okay? And now, a phospholipid itself is made up of two things. It's made up of a phosphate and a lipid. And as a consequence of that, we have a head, which is what we call hydrophilic, and I'll highlight that word here, hydrophilic head. And what this means is it water-loving, likes water, isn't deterred by it. On the other side of things, though, we have these hydrophobic tails. And these hydrophobic tails are the opposite. They're scared of water, okay? So they're water-hating. So when we're looking at the extracellular fluid, so the fluid outside the cell, compared to the cytoplasm, so that jelly-like substance on the inside of the cell, we notice that the phospholipids have arranged themselves in something known as a bilayer. Now, the reason for that is that the hydrophilic heads, so these things which I'll circle in orange now, so here's one hydrophilic head, here's a hydrophilic head on the opposite side. These heads face out towards the water. So we know that the cytoplasm is made up of this jelly, but there's quite a lot of water here. And on the outside of our cell, in living things on Earth, we find that we need lots of water. And so both of these fluids have water, but but the tails really don't like water. They're made up of fat, and if you've ever seen oil and water, they generally don't mix very well together. So the heads point outwards and the tails point inwards. And because it takes two to tango in this situation, we end up with a bilayer. We end up with 2 rows of phospholipids with the heads pointing outwards and the tails pointing inwards. So all of these phospholipids are arranged in that, keep it consistent, in that bilayer, where the head of the phospholipid is hydrophilic and the tail of the phospholipid is hydrophobic, okay? And these molecules, they spontaneously, they form this bilayer where the tail faces inwards and the heads face outwards, simply because of the composition that we just spoke about of the extracellular and intracellular fluid having a high water composition. Now, our cell membrane is not just made up of these phospholipids, right? We saw in our diagrams earlier that we've got lots of other pieces, so let's gradually fill in the puzzle now. So what I've got, and I'll make them a little bit clearer in maybe some orange here, there we go, is cholesterol. You've probably heard of cholesterol before. We do have good and bad cholesterol, and in this instance, this cholesterol is good for our cell membrane, okay? Now, these cholesterols, as you can see, I've highlighted, we've actually got another one poking in on the left there. They are maintained in between the phospholipids. They're not on the inside or outside, they're in between those phospholipids, in between those phospholipid tails. Now they maintain the fluidity, that flow, the ability to move and maintain its structure. That's cholesterol's job here, really nice and simply. Up next, in green, I've got carbohydrates. Now, carbohydrates are found on the exterior surface of the membrane. So they're either going to be attached to phospholipid heads, like I'm highlighting in green here, or attached to a protein, okay? And what they do is they act as markers. And what they're going to do as a marker is they not only identify this particular cell, like, hi, I'm a red blood cell, or they can also detect and recognize harmful cells, so cells which are pathogenic, right? So carbohydrates, they're found on the outside. The reason that they're found on the outside is they're like a characteristic of that cell. If they were found on the inside, well, they wouldn't be seen and they wouldn't be any good. Up next, and finally, we have a couple of different types of proteins. Now, I'm just going to gloss over them today because they span the entire length of the phospholipid bilayer, or the entire length of the cell membrane, but we've got 3 distinct types and we'll talk about them as we talk about transport through the cell membrane. So our first kind, really simply, is a channel protein. A channel protein is something that transports ions. Secondly, we've also got a carrier protein, and this one is what transports larger molecules generally, although they can transport ions as well in some examples that we'll look at. And the final kind that we look at, and this is not an exhaustive list, right, is receptor proteins. And again, a receptor in the terms of this cell is something that allows for communication with the external environment of the cell. So we can see here, if I get my pink pen out, oh, that's no good. This is my channel protein on the left. We can see the red arrow running through the center because it's acting like a channel through the phospholipid bilayer itself. And then to the left of that, again, blue, not a great choice of colour on my part, we have the carrier protein. And we'll talk about the action of that in a second. So what we'll do very quickly now is we'll just scroll back up to this image that we glossed over earlier, and we'll just label some of these key components. So if we're talking about this cell, what we're going to start with is this item in the top left-hand corner. So I'll put a little red marker here. Now this here is attached to a protein, and we could almost say it's attached to a phospholipid head. This here is our carbohydrate, okay? That's our marker. And what this allows us to do is actually tell which side is the inside versus the outside of the cell. Because carbohydrates are only found on the outside or in the extracellular solution. It means that this stuff down here is going to be our cytoplasm or our intracellular solution. Up next, we have a protein. Now this protein, as we'll dive into a little bit more, it's kind of bound in the middle and it's going to change shape quite dramatically. It's not an easy channel, so we're just going to identify this as a protein for now. Later on down the track though, what we should be able to identify is that this is a carrier protein. Top right hand corner, look, identical structure attached to a protein found on the extracellular solution, it must be a carbohydrate. And just below that, we're spanning the entire width of the cell membrane here. Now this here, we could refer to as the bilayer, but we're going to be a little more specific here, and we're going to refer to this as the phospholipid bilayer. Okay. Now, we're only missing one key component here, and this is the structure that helps maintain the fluidity of the cell membrane, and it's no other than cholesterol. So that's all the components labelled there. The only thing that I've pre-labelled for you is the individual phospholipid molecules themselves. So again, we've got that hydrophilic end, the water-loving end, and we've also got the hydrophobic end, water-repelling. So if I colour that in on my cell membrane, we could see that all of these tails really don't want the water found in the extracellular solution of the cytoplasm, but the heads, they don't mind that. Okay, and these heads span the entire duration of that cell membrane.